Okay, so we're looking at the structure of DNA and RNA. So inside the cell and inside the nucleus, the DNA comes in two forms, chromosome form and chromatin form. So let's take a look at some electron micrographs of DNA in these two forms. So let me get my pen here. So this is an electron micrograph of DNA in chromatin form, and this is an okay, so we're looking at the structure of DNA and RNA. So inside of the cell and inside the nucleus, the DNA can take two forms, chromatin form and chromosome form. So let's take a look at some electron micrographs of each different form. So let me get my pen here. So this electron micrograph is of DNA molecules in chromatin form. And this electron micrograph is of DNA in chromosome form. And the difference is that this long strand, many strands of DNA here, the long strand of DNA gets folded up and wrapped around proteins into the chromosome form. So one single human DNA, linear DNA, can be as long as 2 meters. And so to get wrapped up into a nucleus of a cell, it gets wrapped around proteins. So here's our cell and our nucleus for a eukaryote and for a prokaryote. So here it shows in this cartoon the DNA in chromatin form. And remember, our eukaryotes, the DNA is in long strands, so it's got an end on either side. In prokaryotes, the DNA, there's one DNA and it's in circular form. So let's take a look. Eukaryotes, let's take a look at the two groups in eukaryotes. So we've got bacteria and archaea. So in eukaryotes, the DNA, the chromatin, the DNA molecule gets wrapped around what's called a histone protein. Bacteria, the circular DNA gets supercoiled and proteins are used to facilitate that supercoiling, but bacteria don't use histone proteins. What's interesting is that archaea do use histone proteins. It's still one circular DNA, but they use histone proteins to wrap up the DNA. And that's why archaea is thought to be more closely related to eukaryotes than to bacteria, because both archaea and eukaryotes use this histone protein to wrap up the DNA. So let's take a look. What does that look like, having DNA wrapped up? So here's our DNA, and it's in a double helix. So remember we've got, let me get my pen. Remember we've got our 2 strands of nucleic acids that are attached together with these hydrogen bonds. So that DNA molecule then gets wrapped around this histone protein. And that's one of these right here. So that is called a nucleosome. And it's kind of like a bead. And then it gets coiled again. So it's almost like a chain with beads on it. And then that necklace of beads gets coiled into a fiber. So this is still considered our chromatin. So there is some wrapping in our chromatin form, but then to become our chromosome form, there's even more wrapping. It's called condensation around another protein. And you can see, so this chain right here then gets wrapped around this other protein to form our chromosome. And since it's coiled so tight and wrapped so tight, we can now see it with a light microscope. So what you're seeing right here, this is a form that we normally draw our chromosomes in this X shape. And what this is, there are 2 chromosomes here, and it's a duplicated chromosome. So this is one chromosome right here, and this is another chromosome, and they're exactly alike. So this chromosome has been duplicated to this one right here. So there's actually 2 chromosomes right here. Okay, so why 2 different forms? Well, if you think about it, let's go back to our picture. This form right here, our chromatin form, it's much easier for proteins that are needed to replicate the DNA to be able to get in here and get access to the DNA molecule and to our nucleotides. Let me get my pen here. In this form right here, where our DNA is folded and wrapped up, it would be very difficult for proteins to get in here and replicate the DNA. So it's easier to be in chromatin form for replication. It's easier in chromosome form to move the chromosomes around inside the nucleus and inside the cell. So during division of the cell, when you need to get replicated DNA into different parts of the cell so that the cell can then divide in 2, it's easier to be in chromosome form. And I think of it kind of like a ball of yarn. So if I need to transport some yarn, would it be, let's say I've got 5 different colors of yarn that I need to transport around. Would it be easier for me to transport them around all the string or yarn all unraveled? Or would it be easier for me to wrap each around in a ball and then be able to carry each ball around. I think it'd be easier to carry yarn as a ball or all wrapped up. But when you actually have to use the yarn to knit or to sew thread, it's easier to unravel it. So that's the way I kind of think of it. Okay, so I hope you found that interesting. And next we're going to talk about the difference between DNA and RNA.